Assalamu alaikum. My name is Khalid Alnajjar. I'm a second year. Those are my contact information. In case you want any question, feel free to contact me about this lecture, about any other lecture, any other course. Feel free to contact me. So I'll be discussing in this lecture. This lecture is about the disposal of amino acids or nitrogen mainly. It's about degradation or how to get rid of nitrogen. Those are the objectives. You can go over them quickly. Like quick overview over the lecture. It will be talking about how to break down amino acids and getting rid of them. I know some of you want the amino acids to stay in their body, but no, you don't want this because amino acids are toxic, and for a couple of reasons that I'll be discussing now, inshallah. Amino acids don't have storages in our body. We don't have a storage for amino acids in our body or proteins generally, unlike carbohydrates and fats. You know how carbohydrates are stored as glycogen, and fats are stored as lipids and so on, adipose tissue. You can't store amino acids. And if amino acids were to stay in your body, they would be very toxic to your brain, and they will just, diseases and syndromes are related to problems in degrading amino acids and the fact that they stay in your body. So they are very toxic and they cause toxicities and so on. Generally speaking, there are two phases for degrading amino acids. The first one is just breaking it down and forming an amino group and forming an alpha keto acid, which will be a form of energy. I'll be talking about all of this soon, inshallah. The second phase is using this alpha keto acid as a form of energy, or in other words, a form where your body can deal with. You know, because your body can use your muscles or your proteins, or less simple, the amino acids for energy, not only glucose and fats. Okay, you can see here this second. Yeah, you can see here this figure is just to show you what the protein turnover mainly. Yeah, there is one important point to mention here. You know, amino acids are, since there are no storages to store amino acids, the only way to get amino acids is either through synthesis or de novo, or obtained through diet and food. And you excrete them, you don't store them, as I mentioned, through something called urea and ammonia. I'll be talking about them throughout the lecture, inshallah. Nitrogen, you know, as nitrogen is a very basic molecule in an amino acid, and a healthy human being has a balance of nitrogen in their body. There are two terms that you need to know here: a positive nitrogen balance and the negative nitrogen balance. First of all, before that, what do I mean when I say a healthy person has a balance of nitrogen in their bodies? It's a balance in which the excretion equals the production or the synthesis and so on. It's written here, by the way. Healthy, well-fed individuals are in nitrogen balance, in which nitrogen input equals output. Yes, this summarizes excretion and storage. No, not storage, sorry. Excretion and synthesis and diet. They have to be in balance. So if you increase, for example, if you increase your protein intake, you have to increase, or your body has to increase the nitrogen output or excretion just for the sake of this balance. So when I say a positive nitrogen balance, or a person is in a positive nitrogen balance, this means your body is storing the nitrogen more than it is excreting. So it's positive. What are the examples of cases of people which were with positive nitrogen balance? You see here, childhood or pregnancy, or someone recovering from an illness or trauma, or so on. So they have more nitrogen in their bodies than they are excreting it, because, for example, in pregnancy, a pregnant woman is using this nitrogen for the developing baby in her body. Or a child is developing and growing up, so they need this nitrogen. Same goes with recovery from an illness. Negative nitrogen balance, on the other hand, is the opposite, when your body is excreting more than it's taking or synthesizing. So output more than input. We have a couple of examples here, like, for example, during stresses or during trauma or burns, illnesses, surgery, injury as well, by the way. Injury is trauma. You see here, if you want to compare them, not to mistake both, this or negative nitrogen balance is during trauma or illness. Positive is during recovery from an illness or a trauma. Okay, don't mistake them. Another example for negative nitrogen balance is during malnutrition. You don't have malnutrition is you don't eat what you're supposed to eat, or you don't eat your intake levels of nutrition. You will have less protein, malnutrition basically. By the way, a question will come about this in the exam, if not two, about the examples and so on. Mark my words. This slide is talking about the amino acid transport. I will be talking about the amino acid transport from the stomach, because throughout the lecture, my main focus will be the transfer from muscles and other tissues to the liver. So just here, mainly stomach. Your intestinal enterocytes has, as you saw in GI, quick flashbacks, you have sodium-dependent secondary active transporters. Okay, and I want you to know that each amino acid or each couple of amino acids have different transporters for them to transport from the stomach or from your diet to your body, to your blood, to the liver, and so on. Okay, know this because there are diseases related to this, as I'll be talking about it now. Yes, here it is. We have cystinuria, for example. Cystinuria is basically a problem or something is wrong with the transporter that transports those amino acids, which are cysteine, ornithine, arginine, and lysine. Okay, and there is a mnemonic which is COL. So what is cystinuria? Cystinuria is the transport system that transports COL. I'd call them COL. Okay, amino acids is defective. Okay, so you will have an inability in absorbing COL amino acids as written here. Okay, COL amino acids again are ornithine, arginine, lysine, and cysteine. And just think with me, if I say your body cannot absorb certain amount of amino acids, or your body cannot absorb X amino acid, then by common sense, this amino acid will appear in urine or in the stool, mainly in urine. As in the case of cystinuria, COL amino acids will appear in urine, as I highlighted it here, because it's very important. It's the main, a scenario will come in your exam, which will be talking about a patient, blah, 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 and so on. And you will spot one sentence in the whole case or in the whole scenario saying lab tests showed COL amino acids, or for example, lab tests showed cysteine in urine, or ornithine in urine, or any one of them, of these enzymes, or all enzymes maybe. Okay, you directly know that this is cystinuria. Okay, you don't need to go into deep details about diseases. You only need to know certain details that are actually important for your level. And when you know that, for example, when you read a scenario which says arginine was found in urine, khalas, this is cystinuria. That's the bit of information you need to know. You also need to know type 1, there are two types, type 1 and type 2. Type 1 usually have normal urinary amino acid profiles. Type 2 and 3, it's written here, and it's self-explanatory. Cystinuria show various degrees of hyperexcretion of cysteine and dibasic amino acids in urine. So you will see those amino acids in urine in type 2 and type 3. Okay. The last point about cystinuria, that those amino acids can make or can precipitate in the kidney and in the urinary tract, forming stones or known as caliculi, which will block the urinary tract. Just a quick summary here. I just don't want to overwhelm you. Well, before I tell you the summary, look at always when you open a slide and study it, just look at the highlighted sentences or highlighted phrases or words. Those that are written in a different color, that are written in a bold, those are usually important information. In other words, the doctor is trying to steer your focus or into this piece of information or into this word, and he or she will be asking you about this word. So make sure you know them well. In this slide, for example, COL, you need to know that cystinuria is a defect in absorbing COL amino acids. They appear in the urine. There are 2 types or 3 types, type 1, 2, and 3. and that they can form stones. The other disease is heart nut disease. There are two diseases in this whole lecture, cystinuria and heart nut disease. The other disease is heart nut disease. Heart nut disease has a similar concept to cystinuria. However, the defect here is in the absorption of tryptophan, not called amino acids, in tryptophan. Okay, so again, without reading the whole slide, if you face a big scenario, a long scenario, and you spot this sentence saying lab tests show lab tests show tryptophan in urine, you directly know that this is heart nut disease. Okay, most probably heart nut disease, because that's what you are needed to know in this level. Okay, there are a couple of points here that you need to know. That tryptophan is a precursor for vitamin B3, which is known as niacin. Okay, as written here, niacin, vitamin B3 is made from tryptophan. So in so niacin or vitamin B3 is synthesized in your bodies, and the process of the synthesis of this crucial and important vitamin requires tryptophan. So if you have a defective absorption of tryptophan, you're not absorbing tryptophan. Tryptophan will go into the urine. You will not have enough tryptophan in your body. You won't be able to synthesize niacin. Okay, hence you will have symptoms, or the manifestation will be symptoms of niacin deficiency, which are called pellagra-like symptoms. What are pellagra-like symptoms? They are usually abbreviated into three. It's a mnemonic. It's called 3Ds, or sometimes 4Ds. Here it's written 3Ds: dermatitis, dementia, diarrhea. The fourth D is usually death, by the way. Dermatitis is, as you can see here, this is called dermatitis. Dermatitis is inflammation of the skin. Itis is inflammation. Derma is skin. The dementia is just forgetting stuff, just as in Alzheimer's, for example. Diarrhea is diarrhea. We all know diarrhea, and we all know death, but death is not written here. Okay, I also highlighted those. By the way, the doctor writes them in red, but I highlighted them because I feel like they are the most important things to know in this slide. Okay, defective absorption of tryptophan. Tryptophan will appear in the urine. Niacin won't be synthesized. Pellagra-like symptoms. What are pellagra-like symptoms? They are the 3Ds: the dermatitis, dementia, and diarrhea. That's basically heart nut disease, or that's basically what you are supposed to know from heart nut disease. Like, this is a checkpoint. This is supposed to be a checkpoint where I ask you, do you have any questions? Did you understand everything? But since this is recorded, I'll just go on. Sorry. This slide is talking about the cellular. Let me read the title, it's better. Cellular proteins are degraded at different rates. What do we mean here? It's just an introduction to what's coming next, or how are the amino acids being digested? How are they being broken down, and so on. It's just, you can read it, by the way. It's pretty straightforward, nothing very important here. And you can tell the doctor didn't highlight or bold any word. But I'm not saying that all the questions are from the highlighted or bolded words, no. But it's just an introduction. Yeah. Introduction to this, basically. Okay, how are amino acids, basically, or proteins, degraded or digested or broken down? Through a system called ubiquitin tagging. What is ubiquitin tagging? Ubiquitin tagging is basically, I'll explain on this figure. This figure makes it really easy. So if there are amino acids or proteins that are damaged, or they are in excess, or we don't need them, or and so on, they have to be degraded. As I mentioned before, we cannot store them. We don't have storages of amino acids, and if they stay, they are toxic. So we need to degrade them, throw them away, basically, excrete them, get rid of them. How do we get rid of them? Or as the question here implies, it's really straightforward. How can a cell distinguish proteins that are meant for degradation and proteins that are meant to be used? Through the ubiquitin tagging system. So ubiquitin comes and sticks to the amino acid that is meant to be destroyed. And this is mainly at the tag in which this amino acid is known to be destroyed, or now your body knows that this amino acid will be destroyed through this tag. Okay. You can see here, by the way, it will be misleading. Look at those black dots. Yeah, this chain with black dots and white bonds. This is the amino acid. The pink thing is the ubiquitin. Yeah, it's big. The C terminus of the ubiquitin will stick to something called epsilon. Let me go to the next slide to make it easier. Epsilon amino group of lysine residues in this case. This is lysine, okay? And this is how it's tagged. You can see here, a small protein presents in all eukaryotic cells is the tag that marks the proteins for destruction. C terminal of the glycine will become covalently attached to the epsilon. This is the Greek letter that is read as epsilon, amino groups of lysine, and this is how you know, or how your body knows that this protein or this amino acid is destined to be degraded. The energy, let me show you here. You see, this is the pink thing is the ubiquitin, and the black thing again is the amino acid. The ubiquitin binds to the amino acid through ATP or isopeptide bonds that come from ATP hydrolysis. Okay? Then now the amino acid is tagged. Okay, it's tagged for death. Where is it digested? In something called the proteasome. Proteasome will come and digest the ubiquinated, as written here, proteins, or the tagged proteins. It's called the proteasome. Yeah, it's 26S proteasome. Now your body will not digest the ubiquitin. It will only digest the amino acid. This is the job of the proteasome. The proteasome will differentiate between the ubiquitin, and it will recycle it, and with the amino acid, and it will digest it. And it will digest it into two groups, an alpha amino group and alpha keto acid. As I mentioned in the beginning of the lecture, the alpha amino group will be excreted, and the alpha keto acid will be used as a source of energy. Alpha amino group is the toxic thing we have and we are afraid of, or your body is trying to get rid of, because if it stayed, it will be toxic. So this is the toxic part of the whole body, and it's called ammonia. Ammonia is toxic to your body. Yes. How do you get rid of this? Yes, perfect. طيب. It's okay. Came back. Okay. طيب. Okay, I will start talking now about the removal or the digestion of the amino acid, which starts with the removal of the alpha amino group or the ammonia. You see, you're not supposed to know structures, by the way, but I will explain on this structure, because I think if you understood it, you can just imagine it and you will understand everything basically. This is an amino acid. It could be any amino acid of the 20 amino or whatever amino acids we have. This N and double H or nitrogen and 2 hydrogen. Let me not overwhelm you with chemistry names. This group is called the amino group, which is basically stands for ammonium. NH2 is ammonium. This group in the middle is called the R group. Let's refer it to, this is the R group, but let's refer to this all as the R group. And this is the carbon group. When your body digests the amino acid, it basically breaks off this and removes the ammonium from the whole amino acid. And then this way, you can produce energy from what's rest, and you can get rid of this toxic part, because as long as this part is free, which is the ammonium, it is toxic. As if it's bound to any other amino acid, it's no more toxic. You get rid, you break this bond, you get rid of this ammonium, and then this you can use it as energy source, or what I've mentioned before, the alpha keto acid, which can be converted to Na to ATP or any other any other form of energy that your body can store and can deal with, basically, because it can deal with a carbon group. Basically, it has to be ATP or glucose, maybe anything. Okay. Yeah, this, what I just explained, is done through two reactions. The first one is transamination. The second one is oxidative deamination. Okay. This is how you break down the ammonium group, or remove the amino group and get rid of it through those two reactions. Okay. Again, here, look at the doctor have written it in red color. NH3, which is ammonium, which is this, is highly toxic and has to be removed by the body either as free ammonia or as urea. You will see now that most of the ammonia in our body are excreted as urea. However, some of it, or chunk part of it, is excreted as ammonia itself. Sorry. Okay. Most of it is excreted as urea, and some of it is excreted as ammonia. Okay. This is basically what I explained. The digested amino and the digested proteins of what we eat will become amino acids, the simpler part of the proteins, which will be degraded in the liver. All of this happens in the liver. Basically, everything happens in the liver. It will be degraded into an NH4 molecule, which is the ammonia, and an alpha keto acid, which will be used as an energy or ATP later. You can read it here. And alpha keto acid will enter the metabolic mainstream as precursors to glucose or citric acid cycle intermediates, which will basically become ATP, because glucose will end up as ATP. Citric acid, if any metabolite will enter the citric acid cycle, it will also end up as ATP. And then ammonia will be just excreted or removed from the body. Right. Let's start with the first reaction, which I said here, transamination and oxidative deamination, to remove the ammonia here, or the amino group. It's called the amino group for now. Let's start with transamination, or the first reaction. Right. Transamination is basically removing this amino group and giving it to an alpha keto acid. So let's get back to this. Yeah, I like this. I like this structure. Removing this amino group and giving it to another molecule or another structure that is called an alpha keto acid. Okay, you will see now. This process is called transamination, and it's taken care of through amino transferases, or enzymes called amino transferases. Okay. Here is an example. I'll explain more examples in the next slide. But let's read this. This is good. The alpha amino group of many amino acids is transferred to an alpha ketoglutarate to form a glutamate. This is an example of what I just explained. I just explained the generic complex, or the generic process of transamination, and this is just another example. Okay, so the alpha amino group, this alpha amino group will be taken from this amino acid and given to an alpha keto acid. As I just said, this is the generic. In that example, the alpha keto acid is glutamate. Sorry. It's called alpha ketoglutarate to form glutamate. Yeah, I just messed up. Let's go to the examples and I will explain everything. These are the examples. Now we will understand everything, inshallah. Aspartate, we have two main examples you need to know, or two main transamination processes that you need to know: aspartate amino transferase and alanine amino transferase. For example, alanine amino transferase, the amino acid is alanine. Okay, the alpha keto acid is called the alpha ketoglutarate here. So alanine amino transferase, or by the way, those are the ALT, AST, infamous ALT, AST, enzymes, liver enzymes that are just tested in every other liver disease. Yes, these are the enzymes. So this is ALT and this is AST. ALT will take the alpha amino group in alanine and will give it to alpha ketoglutarate. Okay. Now, alpha ketoglutarate with the alpha amino group will be glutamate, and alanine will be part of it. Okay. Yes, this is it. Glutamate now has the alpha amino group, which is toxic and will be gotten rid of, and I'll explain how in a bit. Part of it is the alpha keto acid, which is used as an as a metabolic intermediate or ATP or any other form of ATP that your body can deal with. And pyruvate is actually a form of ATP or a precursor to ATP, let me say, that your body can deal with. Pyruvate is part of glycolysis. Yes, it's the part of glycolysis, and it is what comes from glucose. And after that, it will complete to the TCA cycle and blah 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 and so on until it forms ATP. So pyruvate is actually a form of ATP that your body can deal with. Okay, glutamate is the carrier of that toxic alpha amino group, and that's how your body will get rid of it. AST also, again, aspartate amino transferase, the same exact formula, but with different ingredients, let me say. Aspartate is amino acid. AST or aspartate amino transferase will take the alpha amino group from aspartate, will give it to alpha ketoglutarate to form oxaloacetate and glutamate. Glutamate is the alpha ketoglutarate plus the alpha amino group, the toxic alpha amino group. Sorry, and oxaloacetate is the ATP intermediate that your body can deal with. Okay, and I also want you to know that these reactions are reversible. Your body can go this way towards glutamate to get rid of the alpha amino group, or the opposite, to form or to synthesize alanine or any other amino acid with any other process. Yeah? Again, this whole slide is just explaining what I said, what I mentioned. Let's read it together. Transamination is catalyzed by the family of enzymes amino transferases or transaminases. They have two names basically. They will take the donor. The donor will be the donor will be the amino acid. It will donate this ammonium. It will give it to the. It's hidden by the camera. Sorry. Yes. This is the enzyme. This is the amino acid with the alpha keto acid, which is alpha ketoglutarate. No, it's from here. This is misleading. Why? This is the amino acid. Okay. This is the toxic part of the amino or the alpha amino group. Okay. This is the alpha ketoglutarate. AST or ALT. This is by the way, this is as a generic example of the whole process. Let's say this is ALT and AST. This will be alanine. This will be alpha ketoglutarate. The enzyme will be ALT. ALT will take this toxic amino group from the amino acid, will give it to alpha ketoglutarate to give us glutamate, and will give pyruvate. The alpha keto acid will be pyruvate. Yes. This way, I explained it this way. Let's get back to the generic part. I want you to just put in your mind the generic process, and whatever example they shoot you with, you'll just directly refer to the generic process, and you'll directly know the answer or understand the whole process. But basically, they will not ask you about anything except ALT and AST. Alpha amino acid, I will explain this again just to make sure that it's very clear. The alpha amino acid, or this is the amino acid. This is the alpha ketoglutarate. Okay, the enzyme will come, which is called amino transferase or transaminase. It will form glutamate and an alpha keto acid. Glutamate is the carrier of the amino group that is toxic to your body and that your body needs to get rid of. The alpha keto acid is the ATP form that your body can deal with. Fair enough? I think this is clear. I'll just, again, I will give it another example just for you to understand. Let's take aspartate. The alpha amino acid will be aspartate. The alpha ketoglutarate will be alpha ketoglutarate because it's just an ingredient that will take the ammonium. The enzyme will be aspartate transaminase or AST. So AST will take the ammonium or the amino group from the amino acid, will give it to ketoglutarate, will give us alpha keto acid, which was oxaloacetate, and the glutamate. Okay, the glutamate will be the carrier of this toxic ammonium, which your body will get rid of, and the alpha keto acid will be oxaloacetate. Oxaloacetate is an intermediate of the TCA cycle, and it is a form of energy. Basically, it's the precursor to ATP. complete in with the process, you will have ATP. Yeah, this is basically transamination, all of transamination. Okay, one more thing you need to know, two main very important things you need to know, and I think they will be asking you about it. Glutamate here, have you not noticed that glutamate is not changing in both? Let me go back to this other slide. Yes, glutamate here is the same glutamate. It's in all transaminases reactions or aminotransferase reactions. Glutamate will be the same and will be a part of the whole process and the whole reaction for any other for any transamination reaction, because it is the carrier, or it is, let me say, it is the safe carrier of nitrogen in your body. Okay, it's you can just imagine it as the taxi of nitrogen. So nitrogen or ammonia, nitrogen is just a part of ammonia. So glutamate will be the taxi that will take ammonia from the amino acid and will be taking care of it in order to excrete it from your body, or and getting rid of it. This is very important. You will get an example of, you will get a question about this. What is a temporary storage of nitrogen? Because remember what I mentioned in the beginning of the lecture, no storage in our bodies for nitrogen. Glutamate is just a temporary storage, because it's just taking the nitrogen or the ammonia from the amino acid and delivering it, I don't want to overwhelm you now. It's just taking it from the amino acid and completing throughout the process of getting rid of this ammonia or this nitrogen. That's why it's just a temporary storage. So it's not like a literal storage or a room where your body just fills in. Like, for example, in fats, adipose tissue is just a place where all the fats and lipids are stored in. No, it's just a temporary storage that will take the ammonia and complete through of the process of getting rid of ammonia. Okay, I remember we got a question about this. That's why I'm emphasizing on it. It's a glutamate. Okay, it's not a glutamine or glutamine. It's a glutamate. Okay, which is glutamic acid. The other very, very important thing about transaminases is that they require a cofactor that is called pyridoxal phosphate or vitamin B6. Vitamin B6 is pyridoxal phosphate, and this whole process will not carry on without pyridoxal phosphate. So if someone has deficiency in pyridoxal phosphate or vitamin B6, he or she will not have transamination in his body. He will not get rid of ammonium, basically. Fair enough, easy. Again, what are the very important stuff you need to know in transamination, or all of it? It's just that the whole process, what does it do? What is the function? What are the enzymes, AST, ALT? They just take the ammonium, give it to alpha ketoglutarate to form a glutamate. Glutamate is the carrier of ammonium. It's just a temporary storage. This is the second most important thing. And then we are left with a form of energy that your body can deal with, and it is called alpha keto acid generically. It can be pyruvate, can be oxaloacetate. The fourth and last one to know is the cofactor, which is written here as PLP, which is pyridoxal phosphate, which is vitamin B6. This slide and the next slide will be basically talking about ALT and AST, but I explained everything about them, but no harm in just reading them or going over them quickly. He's here telling you that ALT stands for alanine transaminase. Okay, one thing in this slide that I didn't mention, that you know how I told you that the two main examples of transamination is ALT and AST. So most aminotransferases or most transaminases or most of the transamination processes will work in the direction of a glutamate. Okay, let me make this very clear for you guys. This, I told you here that they are reversible, and they can go in the direction of a glutamate to give the ammonium to glutamate, or to form glutamate basically, which has the ammonium, to get rid of the ammonium, or they can be vice versa. They can form, or they can synthesize the amino acid from glutamate. I said this, I mentioned this. I just want you to know that most of the aminotransferase reactions will be by default moving toward the direction of a glutamate. except for aspartate. Its default is moving toward aspartate or toward the amino acid, okay? This is the only thing that that slide is trying to tell you. I just want to explain on this slide for the sake of making it more clear and just visualizing it. Okay, so again, most of the amino transferase reactions are held in the direction of glutamate, which is this direction, okay? Except aspartate, by default, it's directed toward forming aspartate from glutamate. So we have glutamate, we have oxaloacetate, we have AST, which is aspartate aminotransferase. AST will take the alpha keto, will take the ammonium, will give it to oxaloacetate. Oxaloacetate plus the ammonium group will give us aspartate. And the glutamate without the ammonium group will be alpha ketoglutarate. So this is basically this reaction in the direction of aspartate, okay? But whatever I explained now works on any other reaction if it's reversible. Okay, I explained from alanine to glutamate, and now I explained from glutamate to aspartate. I just want you to know and make sure that you know well, the most aminotransferases will work in the direction of glutamate or in the direction to form glutamate, okay? So glutamate is basically the collector or the carrier of nitrogen. Except AST, which is aspartate transaminase, it's the exception for this rule. It transfers amino groups, which is the ammonium, from glutamate to oxaloacetate, forming aspartate, okay, as I explained in the previous slide, which is used as the nitrogen source in the urea cycle. I hope this makes sense. This slide is trying to tell you that AST and AFT are elevated in nearly all liver diseases. I mentioned that, and we use them to test for liver diseases, basically. This is a checkpoint for you to ask questions, but again, sorry. Let's go on. Second. Right. The second process is called oxidative deamination. Remember how I told you removing the amino group from the amino acid requires two reactions, transamination and oxidative deamination. We talked about transamination. Now is the time to talk about oxidative deamination. Right. Oxidative deamination is simply, it's very simple. It's literally one or two slides maybe. It's simply taking the ammonium group from glutamate and just giving it to the urea cycle. Okay, what is urea cycle? What is going on? I'll just simplify it all. But this is oxidative deamination in a nutshell. Taking the amino group from glutamate, which, where did we get glutamate from? We get it from the transamination, okay? And giving this ammonium to urea cycle. It is catalyzed by which enzyme? Glutamate dehydrogenase. This is basically oxidative deamination. So it yields to the production of free ammonia. Okay, why? We said that free ammonia is toxic, but free ammonia will go to the urea cycle. Urea cycle is basically a whole cycle just to get rid of ammonia in any form, okay? It will be explained further and discussed further by my colleague, Khalid, inshallah, but this is what the urea cycle basically is. Another important thing about oxidative deamination, it requires NAD or NADP, okay? So a cofactor for glutamate dehydrogenase is NAD or NADP plus, of course. Remember how pyridoxal phosphate or vitamin B6 was a cofactor for any transaminase reaction or any transaminase enzyme, or basically AFT and AST. NAD and NADP, NAD or NADP are cofactors that are required for glutamate dehydrogenase in order for glutamate dehydrogenase to take the ammonium from glutamate and give it to the urea cycle. Let's go over the text here. Oxidative deamination is catalyzed by glutamate dehydrogenase, results in production of free ammonia. As I said here, free ammonia, glutamate dehydrogenase from glutamate. Glutamate dehydrogenase, from glutamate, which we got from the transamination. I want this link to be in your heads, guys, okay? It's very important. We have transamination. That where we have, from transamination, we got glutamate, which has the ammonium group in it, and now oxidative deamination will take this ammonium and give it to the urea cycle in order to get rid of it. This is the only enzyme known that has specificity for both cofactors. Okay. Glutamate is unique in undergoing rapid oxidative deamination. Again, okay. I mean, it's easy, so it's all self-explanatory. The most important thing is what I explained. The most important thing in this slide is what I gave you guys. Glutamate from transamination has the ammonia. Glutamate dehydrogenase will take this ammonia, will give it to the urea cycle in order to get rid of this ammonia. This reaction is called oxidative deamination, and this enzyme, which is glutamate dehydrogenase, requires two cofactors or one of two cofactors in ADP or in AD. Like, this is basically what oxidative deamination is. You see, it's only one slide. That's how easy it is. Yeah. This is, you see, I remember how I told you I want this link to stick in your head, which is the link between transamination and oxidative deamination. Yes, this slide summarizes it all. By the way, we're over with the lecture. Khalas, 2 more slides, inshallah. So, where do you want to start? Let's start in whatever I explained just 2 minutes ago. We start with transamination. What do we get from transamination? We get 2 things. Glutamate, which is the carrier of ammonium, okay, or it's here. We get 2 things. One, glutamate, okay, which is the carrier of ammonium, and an alpha keto acid, which is an ATP intermediate that your body can deal with. Those are the results of transamination, okay? Oxidative deamination is taking the ammonia that is inside the glutamate, okay, that we got from transamination, and removing the ammonia from it and giving it to the urea cycle. This is oxidative deamination. It is catalyzed by glutamate dehydrogenase. And the glutamate dehydrogenase requires NADP or NAD, okay? And remember how I told you in when I was explaining transamination, remember the whole example of AFT or AST. It's the amino acid. Just remember the generic with me. It's the amino acid plus the alpha ketoglutarate. We have the enzyme, which is called amino transferase. The enzyme will take the ammonium, will give it to the alpha ketoglutarate to form glutamate. صح? Now we have a glutamate, which is basically the ammonium plus the alpha ketoglutarate. Remember? So now in oxidative deamination, we remove the ammonium from glutamate. What will we have? Alpha ketoglutarate. Yes, alpha ketoglutarate. Let me go back to make this more clear. I want you to memorize it. So here, I think, yes, this is a good slide. Look at alanine, for example, okay? What is the generic one? The generic one is, yes. I don't want a specific example. I want you to memorize the generic. It's also memorize the examples, because the examples are what is supposed to, what you will be asked about, plus the generic is for both. The amino acid and the alpha ketoglutarate, the enzyme, which is the amino transferase, the alpha keto acid and the glutamate. So we have an amino acid with the ammonium. The amino transferase will take the ammonium, will give it to alpha ketoglutarate to give us the glutamate with the ammonium. Now, this is transamination. Oxidative deamination will come and will take out this ammonium, okay, and we'll just get rid of it. Now, glutamate without ammonium is alpha ketoglutarate, yes, and alpha ketoglutarate and alpha ketoglutarate with the ammonium will be glutamate. You get it? You get my point here? So, alpha ketoglutarate, give it an ammonium, it will be a glutamate, okay? If you remove the ammonium from the glutamate, it will become alpha ketoglutarate again. This is basically it. This figure, this small graph, links both reactions together, and this is what I want you to keep in mind, because those two reactions are what will break down an amino acid. Okay, so if you were asked how are amino acids digested or not digested, let's not say digested, how are amino acids degraded or just excreted from your body, you say we have an amino acid. You have the first process, which is called transamination, will remove the ammonium from the amino acid, will give it to alpha ketoglutarate to form glutamate. Then glutamate will carry on to the other reaction, which is called oxidative deamination. And in this process, the ammonium will be removed from glutamate and will be excreted through the urea cycle. That's basically everything about degrading an amino acid. And if you want to ask me, okay, we just got rid of the ammonium itself, where is the rest of the amino acid has gone? Yani, the rest of the amino acid is the alpha keto acid, which is the intermediate of ATP, which your body can deal with. So for the last time, the amino acid will be degraded into two parts. I explained this in the beginning, into ammonium and an alpha keto acid. The ammonium will be added to the glutamate, which will carry on the next process, which is oxidative deamination, in which the ammonium will be removed from glutamate and just excreted through the urea cycle. The other product is the alpha keto acid, which can be pyruvate or oxaloacetate, which are both energy intermediates that your body can deal with. For the last time, transamination is catalyzed by amino transferases, which are ALT and AST. The cofactor required for amino transferase, any amino transferase, will be vitamin B6 or pyridoxal phosphate, abbreviated by PLP. Oxidative deamination is catalyzed by glutamate dehydrogenase. Its cofactor will be NAD or NADP. Okay, here in this is basically the figure that summarizes everything. This slide is basically telling you that when you eat a meal rich in protein, what will happen? Your body will use the whatever it needs from this protein, and the rest will be excreted because we can't store it. Yes. So common sense, after what I explained, for you to know that after a meal rich in protein, your body will have high levels of glutamate. Yes, because it will use whatever it needs from this protein, and the rest will be excreted. How? Before being excreted, it will be in the form of glutamate. Yes or no? Yes. So that's why glutamate levels will be high in your body. Let's go on reading. Reaction proceed in the direction of amino acid degradation, because the reaction of amino acid degradation requires glutamate to be produced first. That's why it will be high in your bodies after a meal rich in protein. When there is low energy in your cell, now this is the reverse. Now this here is talking about the reverse of this whole process. Okay, when there is low energy in the cell, amino acid degradation is high to produce energy from carbon skeleton derived from amino acids. So it will reverse this. Now this whole topic will be talked about in another lecture. I think it's after the urea cycle, most probably. I think it's called amino acid synthesis and degradation, most probably. Yeah, it's how to get energy from an amino acid, but it's basically the reverse of those processes. Okay, for just to get the energy. Oh, no, sorry. I think he's saying, it's when it is low energy, when your body is in low energy, not low amino acid levels. Sorry, I thought it was because there's another point that talks about low amino acid. I think I messed them up. Okay, just to remove any misconception here or any misunderstanding. In case of low amino acid, these processes will be reversed to create amino acids such as alanine and acid. However, in cases of low energy, your body wants energy. So it will break down the amino acids you have to give us alpha keto acid, okay? Because throughout this process, you are not only getting rid of ammonium, you're also having an alpha keto acid, which is a form of energy or an intermediate of ATP that your body can deal with. Remember, I explained all of this, okay? So again, when you are in a low energy state, your body will take the amino acids from your muscles, okay, will put them into this process in order to get the energy from alpha keto acids, which will be pyruvate and oxaloacetate, and to get rid of the toxic part of the amino acid, because if your sole purpose of breaking down amino acids is energy, you don't want NH to pool or ammonium to pool in your blood. It will be toxic. You will get rid of it by default throughout the process. Okay, that's basically it. Okay, this I told you finished. Those, the next two slides are summary slides. This is also can be a summary slide because I mentioned everything, but I want to mention them again because they are very important. And again, you will have questions about them because I remember we got multiple questions about those glutamine, glutamine, glutamate, and those enzymes. You know, they can be misleading. Right. Let's start point by point. There are many points here. Ammonia produced from deamination reaction in most tissues is transported by glutamine. Okay, whatever I explained before, remember how in the beginning, let me go back to the figure so that you will remember. Remember in this slide, I told you this is amino acid transport. It's just, I told you, I literally told you I'll be explaining the transport from stomach to the liver because the rest of the lecture will be focusing on the transport from muscles to liver and from liver to kidney because you get rid of amino acids through kidney, but you break it down in the liver, okay? Now, and the glutamate, alanine, aspartate, all of these are from the muscle to the kidney. This slide is talking about the transfer of amino acids from other tissues other than the muscle. Okay, from the muscle, it will be alanine and aspartate. Alanine, basically, more than aspartate, because I told you aspartate is directed, it's an exception to that rule, and it's directed oppositely toward making more aspartate. So it's mainly alanine from the muscles. But I think it's written here. I will just show it to you now. However, from other tissues of your body, other than the muscle, the transporter will be glutamine. Okay. Now, where is the misconception? Where will the mistakes be made? People will confuse between glutamine and glutamate. Glutamate, we mentioned the glutamate is the carrier of, or the temporary storage of nitrogen throughout the whole disposal process, or two processes. Glutamine is the transportation from most tissues to the liver, okay? To the liver for the breaking down here. Glutamine, I will explain it on the figure 1 second. Glutamine synthetase adds the ammonia to glutamate, forming glutamine. This is how glutamine is formed, basically. Through an enzyme called the glutamine synthetase, it will add the ammonia to glutamate, forming glutamine. Yes, I know glutamate already has ammonia, but if you add another ammonia to it, it will be glutamine. Just understanding this basic, these basic words, just understand them this way and then memorize them, because this is basically it. I can just elaborate more. Glutamine synthetase will add an ammonia to glutamate to give us a glutamine, okay? Why? Because why are we doing this? Because glutamine is a non-toxic carrier of ammonia. Remember how ammonia is very toxic in your blood? Glutamine is a non-toxic carrier of ammonia. Okay, then it will go to the kidneys, and then it will be in the kidneys. It's basically liver and kidney. So I'm focusing on the kidney because ammonia is secreted through urine. But in the kidneys, glutamine will be converted to glutamate again, and ammonia. Then ammonia will be gotten rid of, and then glutamate will be converted to an alpha ketoglutarate, and ammonia will be excreted or gotten rid of through the urea cycle. This is basically the whole slide. I will go over it again because I feel like there is I think I didn't explain it well. Let's try to read this figure. This is the muscle. I told you the muscle. Okay. Both of these are muscles, by the way. Nice. So let me to my first point. First point. Alanine is transported from the muscle to the liver or kidney through alanine. Okay. Other tissues and muscles, by the way, will transport ammonia through glutamine to the liver. Okay. Glutamine. Okay. And here alanine. How? Glutamine synthetase, which is this enzyme, will take ammonia, will give it to glutamate to form a glutamine. Why are we doing this? Because we don't want ammonia to flow freely in your blood through your organs because it's very toxic. We want a taxi, or we want a safe or a non-toxic carrier of it to carry it, sorry, and make it safe to transport it through the blood to the liver. Okay, then in the liver, glutamine, this is, by the way, this will be explained in another lecture, but I will help you here, inshallah. Glutamine will reach to the liver, then it will be it will transform again to glutamate through an enzyme called glutaminase. Clear? Inshallah. And then that's how glutamate will form again transamination here. Glutamate, AST, or yes, in this case it's AST because alanine is here. If this was aspartate, it will be AST. This is ALT. We have so basically here, glutamate from here, alanine from here. This process is transamination. Transamination will form glutamate. So we have two sources of glutamate now. Just understand it in this way, in this basic way. We have two sources of glutamate from here and from the amino transferase. Then this glutamate will go through oxidative deamination using this enzyme, which is glutamate dehydrogenase, forming ammonia, which will go through the urea cycle and will be excreted as urea through urine. Okay, this is the urea cycle. Now, what I care about is this part. Okay, again. Okay, this will make it easier. Ammonia is transported from the muscles and from tissues to the liver through two things: alanine and glutamine. Okay. How is glutamine formed? Through an enzyme called the glutamine synthetase. Remember it this way: glutamine synthetase to synthesize glutamine. Okay. How this enzyme glutamine synthetase, as you can see here, will take the ammonia, will give it to glutamate. Okay, that's just another amino acid, another precursor in the muscle. Okay, where this enzyme glutamine synthetase will add ammonia to form glutamine, and this glutamine will go to the liver to form glutamate again. Okay, to be reversed again, because glutamine is the non-toxic carrier of ammonia. Okay, and the glutamate will go to the liver. Okay, pause here for a second. Go to alanine. Alanine. How is alanine forming? Again, through amino transferases. We have an NH3 amino acids, glutamate, alpha-ketoglutarate will give us, we have pyruvate, we have glutamate, which will give through amino transferase, or ALT in this case, will give us an alpha-ketoglutarate and an alanine. Okay, alanine will go to the liver. Alanine will go again through amino transferase. Yes, amino transferase or amino transamination will happen twice. Will go alanine to the liver, transamination again to form a glutamate. Now we have glutamate twice here. Glutamate will go through all of the glutamate will go through the oxidative deamination to form ammonia. We'll go over it once again just to make sure that everyone understood it, and I don't want any confusion here. Right? Let's start here again. we have an ammonia, we have muscle with amino acids. Amino acids have ammonia, or amino acids will lead to ammonia. طيب, ammonia here, after the digestion and degradation of amino acids, we have ammonia. We want to get rid of this ammonia. Ammonia will be added to ammonia can be added to glutamate basically. طيب, glutamate and pyruvate comes from glucose in the muscle. طيب. So glutamate, which has the ammonia, and pyruvate will go through the process that is called transamination that we talked about. In this case, the enzyme will be ALT, or alanine transamination, or alanine transaminase, to form alanine and alpha ketoglutarate. Now, alanine has the ammonia, has this ammonia. It wasn't the glutamate. The enzyme took it from here, added it to the pyruvate to give us alanine, and then the glutamate without the ammonia will be alpha ketoglutarate, as I mentioned before. Okay, this alanine will go to the muscle. Sorry, will go to the liver from the muscle. Okay, alanine here will go to the muscle. Then it will go through amino transamination again. You might ask me, why is our body doing amino transferation twice? It's because you can't have NH3 or ammonia flowing freely in your blood because it's very toxic. So you have to take it, put it into glutamate, put it into alanine, which is a safe carrier of alanine. It's one of the safe carriers of ammonia. Okay, then the alanine will transfer through the blood, to the liver. It will go through amino transaminase. Sorry, it will go through transamination through the enzyme amino transaminase, or in this case ALT, because it's all alanine. It will go through the transamination to give us a glutamate and pyruvate. We'll go over it again. Alanine, we have alpha ketoglutarate. ALT will come, take the ammonia that is in the alanine, give it to alpha ketoglutarate to give us glutamate. What's rest from alanine will be pyruvate again, which will go to the muscle again. Okay. This glutamate will go through the oxidative deamination we talked about to form NH3 and go to the urea cycle. This part here is talking about forming glutamine. This is how do we form glutamine from glutamate? How? Through the enzyme glutamine synthetase. Glutamine synthetase will take the ammonia in the muscle or in any tissue in the body. Sorry. Glutamine synthetase will take the ammonia in any tissue in the body or in the muscle, will give it to the glutamate, okay, to form glutamine. Okay, don't confuse them. Glutamine synthetase will take the ammonia, stick it to the glutamate to form glutamine. Why? Because we cannot afford ammonia flowing freely in the blood again. So we have to give it to glutamate and then give it back to glutamine in order to go to the liver, and then glutamine will change back to glutamate. Okay. I told you, glutamate is a temporary storage of ammonia. We can't have the ammonia flowing freely in the blood, okay? So we have to transform it to glutamate. Then glutamate cannot also go to the liver alone. It has to be glutamine. Then glutamine will take it to the liver, and glutamine must go back through the whole process again, being glutamate again, then changing to ammonia to get rid of it through urea. Same thing in the muscle. NH3 or ammonia can't flow freely in the blood. It has to be changed to glutamate. And glutamate also can't go to the liver. It has to change to alanine, because alanine and glutamine are the safe carriers or taxis, if you want, of ammonia. So alanine will go to the liver. We'll have to reverse the whole process again. Go back to glutamate, then go back to NH3. Okay, so a process happening in the muscle or in any tissue, NH3 through a whole process in order to ride with a safe carrier. This safe carrier will take it to the liver. Then this safe carrier will have to just go through the whole process again to give us ammonia, once back in order to get rid of it in the liver or in the kidney. This happens in both liver and kidney, as you can see it in liver or kidney mitochondria. It's basically the whole slide. This again summarizes everything again. This slide here connects both processes, but this slide shows you all the process in all tissues in the body. This is a very good slide. It's gives you a general picture of it all. I hope I explained it really well, inshallah. Please don't confuse glutamine and the glutamate. Don't confuse the glutamine synthetase to glutamate dehydrogenase. Memorize them well, because I remember we had a couple of questions in the exam about every single one of these misleading terms. Inshallah, they are clear by now. This slide is talking, it's just a repetition. Let's go over it quickly. The glucose alanine. This cycle I talked about in this slide, I think I forgot to mention this, from the muscle to the liver, from any tissue to the liver to the kidney, is called glucose alanine cycle. Yeah, it's written here, glucose alanine cycle. Because glucose, in the muscle, it starts with glucose, then NH3 pyruvate, they are all precursors for NH3 to come and write alanine and go back there. Okay, so it's a glucose alanine cycle. Again, he's saying the glucose alanine cycle is used primarily as mechanism for skeletal muscle to eliminate nitrogen while replenishing its energy supply. Makes sense, yeah? Takes the amino acid, takes out the energy of it as in the form of pyruvate or oxaloacetate, or generically called alpha keto acid. Then the nitrogen will just be excreted through urea. Pyruvate, he's just saying it's pyruvate with alanine. Remember, ALT reaction has pyruvate and the AST reaction has oxaloacetate. Alanine is a safe way to transport ammonia from the muscle to the liver. It's only from the muscle. Glutamine, however, you see here, alanine is from the muscle exclusively. However, glutamine is from most tissues. It's written here, yes, ammonia produced from deamination reaction in most tissues is transported to as glutamine in most tissues, okay? But alanine is for the muscle only. In liver, ALT transfers the amino group from alanine to alpha ketoglutarate. The resulting pyruvate is used in the gluconeogenesis. I think this is it. Yes. This is just, he's trying to show us this figure is exactly the same as this figure. And then finally, this figure relates all the processes I explained to the urea cycle. It's exactly the same figure I explained there, but it's just drawn in other colors. I'll show you here. You see, this is the muscle. We have amino transferases or transamination to give us alanine from the muscle. From the intestines, we just absorb them through the transporters we talked about in the beginning of the lecture. Most tissues here, or sometimes can be muscles, by the way, or ammonium will be transferred through glutamine. Okay, glutamine will go to the liver, alanine will go to the liver, glutamine can go to the kidney directly. Whatever is in the liver will go to the kidney eventually. Okay, alanine will go to the liver and glutamine will also go to the liver. They will go through the cycles we explained, which are the transamination, and this is the transamination or amino transferases, and the oxidative deamination to yield ammonia. Ammonia will enter the urea cycle. Okay, through the urea cycle, it will be excreted as urea to the kidney through urine to urine. That's how you get rid of ammonia. That's how you get rid of ammonia, through urea. Again, urea cycle will be explained. It will be discussed and explained by my colleague Khalid, inshallah. I have questions here. This is the end of our lecture. I have some questions here. I'll go over really quickly, inshallah. I want you to pause the video if you're watching this and stop and try to answer this. Then listen to my explanation and listen to my answer. Okay. What is the factor required for all transaminase reactions or ALT and AST? It will be pyridoxal phosphate vitamin B6. This is pure memorization. Remember the slide? Let me go really quick. PAP. pyridoxal phosphate of vitamin B6. It's written here. All transaminase rely on cofactor called pyridoxal phosphate of vitamin B6. Next question. How is ammonia transported? How is ammonia transported from body tissues other than the muscles to the liver? To which one of these? Glutamine. Yes, glutamine. Let me go over all the choices quickly. Glutamate is not how ammonia is transported in the blood, yet glutamate is the temporary storage of ammonia until glutamate becomes a glutamine. And that's the temporary storage. What do I mean by temporary? I mean until that it's only the time in which the ammonia rides with the glutamine. Yeah, just understand in this basic way. So that the glutamine can transport through the blood to the liver, and in the liver, it can come back to become glutamate again, become NH3 again, then enters the urea cycle. It's not alanine, because I said alanine is exclusive for muscles, and here I said other than the muscles. And also it can be aspartate, because aspartate is not a transporter of ammonia. So it's glutamine, it's D. Which enzyme carries out oxidative deamination? It's glutamate dehydrogenase. Let me go over the choices so you won't confuse them. ALT, or alanine transaminase, is the enzyme that catalyzes the transamination reactions. Glutamine synthetase is the enzyme that gives ammonia to glutamate to synthesize the glutamine, hence called the glutamine synthetase. Okay, this is a process alone, this is a reaction alone. Okay, oxidative deamination is carried out by glutamate dehydrogenase, which is D. Okay? Glucose 6-phosphate dehydrogenase doesn't make sense because it's not related to amino acids. It's all in the glycolysis and gluconeogenesis. It's in gluconeogenesis. Last question. Which cofactor is required for oxidative deamination? It's oxidative deamination is NAD or NADP, which is D. You want to go? I won't go over the choices here. Similar to the first question I answered, because it can be misleading. It's pure memorization. You have to know that oxidative deamination is relies on the cofactor for oxidative deamination. It is catalyzed by the enzyme glutamate dehydrogenase, as I said here, and glutamate dehydrogenase relies on the cofactors NAD or NADP. Same with the first question we solved here. Transaminases, which are ALT and AST. Transamination reactions are catalyzed by the enzymes transaminases or ALT and AST. ALT and AST rely on the cofactor and pyridoxal phosphate, which is vitamin B6. I think this is the end of my lecture. I hope you benefited from it. I hope I made it easier. Inshallah, it will be an easy lecture. And again, if you have any question, you can find my contact information here. Never hesitate to ask me. Thank you very much. Good luck.